PNAs, nature ecology, and evolutions. His work has been cited well above 12,000 times with an Edge index of 58. So now you can listen to the keynote speech, which is a pre-recorded speech. However, Professor Singh is with us online on Google Meet.
I hope you have a good review meetings today. First of all, let me start with thanking the organizers to invite me and give me this opportunity to come and talk to you about some of the research work we do. In today's presentations, I'm going to talk about the microbial regulation of terrestrial ecosystem functions, and I will try to provide evidence from the microcosms level to the global scale. But before I go to the presentations, let me start with acknowledging the funding that comes from the Australian Research Council, but also European Research Council, European Union, and some of the industry funding. On the top of my slides, these are my international research partners with whom I collaborate very strongly. First is Dr. Manuel Delgado Beccarizo. He was a postdoc in my lab, now it's moved to Spain as independent researchers. Chandra Trivedi was a PhD student, now he's a postdoc in Colorado State University. Peter Reich is a resident professor in Minnesota University, USA. Dr. Fernando Mestres is a professor in distinguished researchers in the University of Alicante, Spain. And Professor Noel Ferreira is at Colorado University. And on the bottom of the slides, the number of PhD and the postdocs in my lab who has contributed to the work I'm presenting today. The presentations, let's start with a very topics, why healthy soils, and the soil function is important. Human civilizations has risen and fallen on the quality of their soils, or the health of their soils. There's enough evidence to suggest the Mesopotamian, the Indus Valley civilizations, and other civilizations has a direct link with the health of the soil. And for me, there was no better comments made by any politicians than the past president of the United States of America, President Franklin Roosevelt, who said that the nation that's destroyed its soils, destroyed itself. And these statements are correct because without soil functions, terrestrial life is not possible. For human survival, the soils provide the crop productivity, crop productions. It also has a huge role in filtering and the buffering of the water systems for the drinking water, for the irrigations, and the recreational purposes. Soil plays an enormous role in the climate regulations by the emissions and mitigations of the greenhouse gas. And these functions in the soils are carried out by the soil biota, mainly by the soil microorganisms. But we are not talking something that is new. People know the role of biology, importance of the role of biology in the soil functions for prehistoric times. It is mentioned in the oldest human literatures available. One of the best examples is that before the Christ, Cleopatra, when she was a queen of the Egypt, she declared the earthworm to be sacred in the recognition of his contribution to soil fertility. Removal of the earthworm from the Egypt was punishable by death, and farmers were not even allowed to touch them in fear to offend the god of fertility. But obviously at that time people did not know that in fact most of the soil function isn't carried out by the earthworm. It does do some functions, but most of the soil functions is carried by microbial communities, which is what I call the unseen majority. They are most diverse organism on the planet, hundreds of millions of species, and billions of cells within centimeter cube of soils, fungi, thousands of species, and hundreds of thousands of species in few grams of the soils, and so on and so forth. In today's presentations, I'm going to talk about mainly two things. The microbial diversity drivers and the predictions. And the second thing, we'll talk about the relationship between the microbial diversity and ecosystem functions. And in the last few slides, I will mainly talk about what we need to do in the next 5-10 years to address these two questions in a more systematic way. So let us start with the driver of the microbial community structures and functions and compositions. There's a wonderful paper published in 2018 by Professor Noah Ferreira in Nature Climate Change, where he summarizes the literatures to look at what are the key drivers of microbial diversity and community compositions. Not surprisingly, the soil pH, organic carbon, quality and quantity both, and the soil oxygen availability, where it comes at the top, followed by the soil moisture and nutrients. But if you look through the literatures, you will find that there was a very few study that has more than 50% of variation in microbial community explained. So if, for example, there's two of the papers that comes from my group. One is in the PNS, where we look at the global driver. Only 39% variation in the microbial communities was explained by almost 26 environmental variables with measures, including the plant community and diversity. And another papers where we had 58 environmental variables, soil pH, chemistry, particle size, climatic conditions, and so on and so forth. And only 57% of variability was explained in that regional Scotland national database. And so if you look at that, between 40 to 60% of variations in the microbial community are still unexplained. So we started thinking about what can be the other factors other than the soil factors, other than the climatic factors, other than the biotic factors that can also help in explaining the microbial community distribution. One of the things we look at the paleoclimate. And surprisingly to us, the paleoclimate explains something between up to 20% of unique variation in the soil microbial community at the global scale. And someone, including ourselves, initially was baffled why a organisms or a population of the community which have a reproductive time of doubling time is between half an hour to a few days will be affected by paleoclimate, which is 5,000 to 10,000 years ago, what happened to the Earth climate. But what we found is that those abrupt changes in the climate changes the structure of the soils, which have the legacy impact until today. But also the recent findings suggest that the soil organic carbon, even in the top 10 centimeter soils, had a half-life of almost like 5,000 years. So it tells you that those carbons survives over the millennium, and that then had an impact on the microbial communities of today. We also look at the plant diversity, plant traits, and so on the impact of the microbial community, and can they explain a certain unique proportion of microbial diversity. And we come back again with something between 10 to 12%, if I remember correctly, was uniquely explained by the plant traits and plant diversity. And that's sampling was across Australia and the UK. So those are the unique targets we targeted for the variability, for the factors that can explain the distributions of the microbial communities. One of the things we have always been intrigued about the ecological principle. We as ecologists have learned a lot from the plant and animal ecology because that is where most of the ecological theory were developed. The plant ecology, one of and so just before I go there, one of my strong feeling always have been, if ecological principle is true, it has to be true across the different domain of the organisms. So it can't be the ecology of plant will be different from the ecology of animal. There will be changes, there will be shifts in the trajectory of that relationship, but the direction of the relationship should be similar. So if you look at the plant and animal ecology, there are hundreds and thousands of millions of species globally, but this is only few species that dominate across the globe for the plant species. For example, the best example in Australia, we have there's a huge continental scale, a large area, but the plant is dominated by eucalyptus species. Obviously, there's a different 800 species of eucalyptus, but it is eucalyptus genus that is dominant across this Australian area. So we wanted to know, and so this is true across the globe for other things as well. So we wanted to know if this things is also true for the microorganism. So is there, we know there are millions and millions of species, but is there a few species that dominate across the globe, the microbial communities? To do that, we sampled around 235 sites across the globe to represent the different biomes, and we measured a number of soil abiotic properties like soil organic carbon, nitrogen, phosphorus, pH structure, and so on. We also downloaded the climatic data, and we also measured the net family productivity. And then we sequenced for the people with non-biological background. I'm going to use two terms here, the amplicon sequencing and the shotgun sequencing. The amplicon sequencing tells us the number of species and the composition of species, while the shotgun sequencing tell us that tell us not only the number of species but also their functional capabilities, what sort of functions that communities are able to carry out in that systems. So initially we measured the bacterial diversity in the systems, and how did we define the dominant taxa? We generated the taxa informations just for non-microbial experts. We usually do not use the word species in the bacterial communities because the species concept is different, and it's especially having developed for plant and animals on the basis of their ability to reproduce together. Most of the bacterial bacterial reproduction is asexual. And so that does not fit the definition of the concept, so we will use the word, I'll use the horticulture taxa. So how do we define the dominant taxa? We define that is a present among the top 10% of relative abundance of all this taxa we have generated from the global soil that we collected from 235 sites.
I see you driving in Dangola on my way. No.
Sorry?
Were you in Dangola at 9 o'clock? No, not
tonight.
Today?
Yeah.
9 o'clock.
You mean tonight? Yeah, 9 o'clock.
Today morning. When you're coming, you came early.
I came early.
Somebody like you, I don't know what you're doing.
Species or global taxa, only 2% classified as a dominant taxa. So it is coming as something around 500 species taxa. But if you look at the relative abundance of those 2% taxa, dominant taxa, it is a 41%. So it fits into the dominant taxa. That is how it is defined for the plant and animal. So the dominant taxa does exist. microbial communities. And then we went into look into their environmental preferences, and some of them are like this high pH, some of them like low pH, some were more dominant in the dry land, some was more dominant in the high productive soils, some in the low productive soils. And that's to allow us to map out the distribution of this dominant taxa across the globe based on the soil pH map and based on the global aridity. This is on the right side of your slide. If you, in the left side is a dominant taxa, which was dominant in high pH soils or the high aridity soils. And you can see that is almost mirror to each other, suggesting that we can predict the distributions of the microbial communities or a taxa, dominant taxa, based on the preference of their habitats. And this is just for the information that only 20% of that taxas have genomes, only 45% have among the dominant taxa, only 45% have a representative, 29% have a representative strains, saying that our global database is quite weak. This is all the genomes, all the microbial communities available across the globe in the culture. So we wanted to know why, for example, in some dominant microbes, more dominant in particular habitats, not others. This is just one example we look at, is the taxa which are preferred very dominant in arid ubiquitous, but very dominant in arid. And what we found that those taxa have a gene which is involved in sodium transport, very high number compared to the others, other taxa. And somebody will ask what is the case, but because the dry have a very high pH, the median pH in the dry is that region, well, is around 8.5. So, and one of the reasons that pH is high is a very high sodium availability. So the microbes have developed adaptations, genomic adaptations, by which they will be able to expel or remove most of the sodium from inside the cells, and that is why they thrive in that areas. And we went on to do the same for the fungal communities. Here, only 0.1% of the taxa fit into the category of the dominant taxa, which accounted for 18% of the relative awareness in the total taxa. Many people ask that why you do the mapping and what is the importance of mapping and so on. Mapping is enormously helpful. This is one of the best examples. One thing is you can predict the distributions of particular community of the taxa. Here, we look at the fungal pathogens, soil-borne fungal pathogens in the global drylands. And on the left side is this is how a distribution of three most dominant, obviously we don't know a lot more than this species, three pathogens. But the example is here, the Fusarium, the Urea, and the Phoma. This is a current distribution on the left sides of those pathogens in the soils. And then what we do, we use what are the factors that drive the distribution. So one of the factors is the mean annual temperature. So then we went on to look at the mean annual temperatures, how the mean annual temperature will increase by 2035, and then we projected what will happen to the dominance of this species across the globe. And as you can see, the more red, the more dominant the species. And you see these three pathogens, dominance across the relative dominance in the globe will dramatically increase by 2035. Obviously, this is a prediction, and people question that, and fair enough. But so what we did, we went down and sampled two sites, one in Spain and one in the USA, which had a 10 years of the climate treatment, warm treatment. And then we measured the dominance of those controls, which has a no warming effect, and the warm plots. You can see there is almost 1 to 4-fold increase in all those pathogens. So our predictions was supported by the experimental evidences. So the take-home message from this one is that few bacterial and fungal taxa in the soil global. Where the phylotypes can be clustered. This is the first global atlas of fungi bacteria. It provided the most wanted list of taxa, which can be studied more, what their function is, how can we harness that function going forward. The second things we are going to talk about, the relationship between the microbial diversity and ecosystem functions. Why it is important? First, the simple rule about biodiversity-ecosystem function doesn't exist. It does exist in plant animal, that microbes follow the same rule. So ecological question. But it is also important to know that if there is a relationship, then we can harness that relationship to improve the biogeochemical and climate model predictions, which have a large uncertainty currently. Or if we can show the relationship, if we show the quantitative relationship, then we can use this information to, in our management and conservation policy, we develop in the future. This is a paper I wrote in Nature Review Microbiology 10 years ago, 2010 exactly, where we try to demonstrate that how understanding the community response is important for increasing the prediction in the models. So here, I've taken an example of soil carbon, which we know is converted into different processes into carbon dioxides. So state 1 of the carbon is then composed of state 2 and 3 and 4. State 1 to 2, we know that is carried out by the soil biota, microorganisms. If you look at the climate model, which is on the top of your slides, climate model says as the global warming happens or the temperature increases, the loss of carbon from soils will increase. This is a fine as long as we assume the community of the microbial community does not changes. But if you look at the literature, 97 to 98% of literature will tell you that the first things happens under the increasing temperature if the microbial community changes. And if the microbial community changes, then the whole control of the system, the regulational system changes. That means the trajectory of that predictions means nothing, honestly. Because if the microbial community changes, a different community has a different practice, as a result, trajectory will change. And this is what we have been arguing for that if we start including microbial community in the climate model, we can increase the predictions models. So what do we know about the relationship between plant diversity and ecosystem functions? There is three sort of relationship is reported in plant diversity ecosystem functions. The first one is what we call the saturating one, that initially as we add a species, the microbial ecosystem function increases. But it then plateaus because 2 species started doing the same thing, 3 species started doing the same thing, so there is no proportional increase. The second is a linear, where each species is considered unique, occupied with new needs. So as long as you keep adding the diversity, you will increase the functionality of the ecosystems. And third thing is called the de-accelerating or catastrophic, where 1 or 2 species are considered vital. And if you remove that, then the whole ecosystem function will collapse. And all these 3 relationships have been reported for the plant community and animal community in different time periods. But does similar relationship exist for the biodiversity ecosystem for the microbial communities? In other words, is the unifying ecosystem also exist for microbial DEF? The first evidence comes a while ago by work done by the Vendor History group in USA, at that time, I think the Belgium, now it's in Switzerland, where they look at the impact of the diversity, microbial diversity, on the ecosystem functions, mainly the performance of the plants. And again, you see he measured a lot of things. On the x-axis is the number of microbial fungi, and the x-axis is the number of functions. What he found is classical and the saturating relationship as you add a species increasing because function increases, but then plateau. Later on, there's another primary work in now becomes evidence that it's not only the species number origins, but also the beta diversity, the compositions of the communities important in the ecosystem functions. In the bacterial community, there was a work done out in 2005 at the Tom Bell Knowledge Imperial College at the time Oxford, where he did is he cultured the microbes and put in a different number from 1 to 50 and measured the ecosystem functions here in terms of the respirations, which is very good proxy of the overall soil functions or ecosystem functions. What he found is that heat relations, as it increases the relation and the diversity, you will see there is a relationship strong. We also did that in the water systems, but in different way. Rather than culturing the microbes and adding a species one by one, because that has its own criticisms, we took the microorganism and diluted down the diversity by the tenfold dilution. So our species found work between 50 to 400 rather than 1 to 50. We still find the relationships in linear. But obviously, these are the microbial studies, and people criticize that this is not a real world relationship. Yes, to test that, we use two databases, again, the global dryland database. And for that, this study, we sampled 78 sites across the global dryland. As you know, it is account for 41% of the global areas. And then in the other sites, we chose the Scotland database that are used to work on game. And that had 179 sites. Why we choose this site? There is a reason. Again, it comes back to the ecological principle. These two sites are really different. Orange and apple, you can say. The dryland have an organic mean organic carbon is 1.79. While the Scotland sites have mean annual carbon is 12.30%. The rainfall, the mean soil pH in Scotland was 4.2. The mean soil temperature soil pH in Dryland was 8.45. So you can say, and the temperature, the annual mean precipitations also entirely opposite to each other. So our goal is that if the ecological principle is same, has to be same in contrasting environments. And then we measure the ecosystem multifunctionality. So what a multifunctionality means here, that we measure the multiple functions: plant productivity, carbon, nitrogen, and phosphorus cycling, nutrient availability, rate of mineralizations, and biomass, and so on. And then you use the average or the different approach of counting the multifunctionality. So averaging that together and look at the relationship between diversity of bacteria and fungi. You see in the both Dryland and Scotland, relationship remain linear in both sides with the diversity of the microbial communities and ecosystem functions. And this was then confirmed by using the structural ecosystem model or using the same approaches. And we've gone not only in these two sites, later on we also did a whole week sample across Australia and look at the relationship between the biodiversity ecosystem functions, again confirmed this by the structural equation model. This is just a step further. This is a paper that comes out in 2020 in Nature Ecology and Evolution, where we not only measure the microbial community, but other organisms like nematodes, protozoa, and so on. And you see that if you include all these organisms, the variation explained in the ecosystem multifunctionality increases because different organisms contribute different level of function in our ecosystems. But no study and no scientific approach is without criticisms. The approach we use in the global and regional database is criticized. These are statistical correlations. Does not mean a causal relationship. And there is inconsistent finding, especially in 28, if you look at the early 2000 publications, 2004-5, some people will say there's a very good relationship, some people will say there's no relationship between microplastic functions. And then there's a concept, there is always the concept of redundancy. So people, there's so many species of bacteria, they talk about millions and millions of species. If you lose a few hundred, how does that get a difference? To test that, we took another look. We go back and we look at the difference in, and so we took a soil samples, what we call is a diversity by dilution to extensive, where we take a soil and we extract all the microbes, extract the soils using the gamma radiation. When the extracted microbes then goes for tenfold dilutions. And once the tenfold dilution is made, then they reintroduce into different bottles in the sterilized soils. So you have soils where you have all the microbes, suppose have a million species. If you have the soils which have a 10 to the minus 6 dimension, should have only 10 species. And then we then allow the microbial community, microbial biomass to recover. Because this is critical. Otherwise, if you not allow microbial biomass to recover, then the impact of diversity will be measuring the impact of biomass. And in the below panel, you see that after 6 months of incubations, our diversity was maintained in those additions, and the microbial biomass on the right side of the panel, you see the graph, using the qPCR, the biomass recovered. And then we go again to do the multifunctionality measurements and look at the diversity of bacteria and fungi. We have two soils, and on the first panel you have a bacteria, and the bottom panel you have fungi. In both soils, A and B relationship is very strong at the multifunctionality level. I'll come to that in a minute. We go into, so remember soil A and soil B. Then we also went down and extracted microbes from this soil of the 6 different species, and we put 1, 2, 4, and 16 different species with combinations and measure the ecosystem function. Again, you see there is a relationship. As the diversity increases, the functions increases. The relationships look different. Soil A and soil B is doing the work because we are coming back to this relationship again. So our result says there is a very strong relationship between diversity and microbial communities and ecosystem functions. Then what was the factor that was driving the inconsistent report of the biodiversity ecosystem function relationship? And we see there was an initial study, there was a lack of appropriate technology. We used to use methodology like ECG and TRLP, which are not good to measure microbial diversity, we know now today. There was also lack of explicit consideration of the biomass to put those studies. They did not allow the soil microbiology to recover. And if you do not allow the soil microbiology to recover, then you are measuring the effect of the loss of biomass rather than the loss of diversity. And then there was some issue with the theoretical considerations, which I come in a minute. So overall, there was a lack of defined conceptual framework of those studies. Let's take the example, critical considerations. Then the diversity of the biodiversity. If you look at the literature, people measure the structural diversity, which includes alpha, beta, gamma, delta, name it. All sort of diversity. They also, other people measure the phylotypic diversity, other measure the functional diversity. So people use their own favorite metrics and then regress with the ecosystem function. And whatever regression they found, they say, oh, our result does not match this. Our result does not match that, even though they measure something very different. So that you can't compare functional diversity with the diversity. They are different measures. As a result, we get a different function and relationship. But then there's other diversity of functions. There's a different sort of functions. The broad functions that is carried out by most of the bacterial or microbial species. For example, degradation of carbon. Most of the species will be able to utilize glucose sucrose in the cellular glucose sucrose in the soil. But then there is specialized functions, say like pesticide nitrification. Only few species have the capability to do that. Again, people will not take these things into considerations when they look at the diversity of functions and relationship. The third thing they did not consider is the ecosystem property itself. What was the initial level of diversity of ecosystems you study, and how that compares with the study other people study. You are comparing your own studies. And then there's another factor that has been taken into consideration was the nutritional status of the systems. Now, come back, I said to remember that, we come back to this slide A and slide B. Here it is. So we done this diversity dilutions of my microbial community. So D1 have the highest diversity in both soils. D5 have the lowest diversity, which is almost 10 to the power minus 6. If you look at the soil A, and the broad function has the soil respiration, so the carbon competition, you see almost no relationship. There is no relationship between the broad functions, soil respiration versus decreasing diversity in the systems. But in the soil B, there's a very strong relationship. Now your question is, why will be that case? The things lies in the availability of labile carbon. In soil A, there's a lot of labile carbon. So the diversity is needed to break the complex carbon. Labile carbon, sucrose, DOSC, and everybody can use it in furnaces. While in the soil B, the labile carbon is low, recalcitrant carbon is high. As a result, more diversity is needed to break those recalcitrant carbon to utilize that. So that's the example one, the broad functions, how we can have different results if you're not looking at the properties of the ecosystem itself. And then if we go back in those two soils and look at the nitrification and methane oxidation, so you see there is a very strong, these are the specific slide functions, and very strong relationship in both soils, because here only few microbial have the ability to do those functions. And if you reduce the stature, but even by a few factors, you have a direct proportional impact on those functions, even if the nutrients supply is high. I hear what I'm remembering. That is the reason we always, or most of the time, we try to use the multifunction, because it combines the different functions. So something we have not able to pick up the relationship for the broad function. If you combine the specialized function and have a multifunctional basis, you will see that the relationship is always linear between the microbial diversity and ecosystem multifunctionality or functionality in the systems. Some of the other things I want to mention before I talk about the perspective, some of the things I think what we need to move away from the approaches we have been developing, not only looking at the microbial ecosystem functions, but also the interactions. Microbes do not live in the soils at all. It interacts with animals, it interacts with the plants. And so it is important that we look at the interaction between microbial diversity, plant diversity, and element diversities, and how that is then influenced by the environmental systems. Once we quantify that, then we should have started looking at other factors that can impact it, including practices, climate change, and ecosystem properties. And this way, we can really push the discipline forward. We have done some preliminary work. This is just an example to look at, where we look at the impact of the plant species and the climate change, and what explains the ecosystem functions on the climate warming. And the data here you see is that the richness of the plant and the climate change does have an impact on the multifunctionality, but not mostly through its impact on the bacterial diversity, which itself is impacted by the protein diversity. So it is interactions between the microbial diversity and the microbial diversity, plant diversity, and animal diversity that's have the most influential impact of the multifunctionality. And in that way, we can expect a lot more variations in multifunctionality rather than just targeting one organization or one domain of the organization. So the take-home message from my perspective is that the microbial diversity is a key predictor of ecosystem multifunctionality from micro to global scale. Loss of microbial diversity will have at least a proportional impact on the ecosystem multifunctionality. And new conceptual framework is needed, which includes all the biotic and abiotic variables of the diversity function ecosystem style to improve our knowledge of the microbial biodiversity ecosystem function relationship. I will leave you with these slides because this is one of the documents I was involved in editing called Global Soil Biodiversity Atlas. If you have interest in the soil biodiversity ecosystem functions, this is wonderful documents to have. It is a free to download from the web page, so there is no cost involved. So you can just download it from the web page. With that, I would like to thank you very much for your time and presence here at Canada. Thank you very much.
Now, I would like to invite our director, Professor Saman Seravira, to thank the keynote speaker.
Good morning, Professor Brad Singh, And I don't think he will be able to connect through here, but he's on YouTube. And it's a great pleasure. It's a wonderful talk, and I have seen that you put a lot together, and we really appreciate your commitment and taking time to join with us in the NIFS Annual Research Congress in 2019. And again, Professor Braj is not a new person. He's already on board with IFS and we're working with us. And let's work together. I don't want to ask any question, but your work is really important for us as well. Like, let's work together as a global partner. And we are already started, and we are happy to do any contribution equally, like we expect your support and contribution to build this area, like we have totally very much neglected area. There's the biodiversity of soil, microbiota. So please, again, thank you very much for your support. Let's plan the future together, work together. Thank you, Braj.
Thank you very much, sir. We are now ready to begin the technical sessions of the Annual Research Review 2019. I would like to invite the chairpersons of the first technical session, Professor Ratnayaka Bandara and Professor Atula Sumudipala, to take their seats at the head table.
Good afternoon. As we are behind time scheduled with our strengths, we start because of the agenda program with the consideration needs to leave as soon as possible. So without any further ado, probably just to present in the introduction, he will talk to you on plant taxonomy and conservation.
Thank you very much, Chairman. So I'm going to present the work done in my project, our project rather, plant taxonomy and conservation. Our main job is actually to manage the NIFS Popum arboretum, which started as a abundant channel and now is a very rich tri-organic forest with a lot of biodiversity. It's high functional diversity, as Dr. Barraj mentioned. So very good. So what are we doing in our project? We are actually trying to answer a few questions. We are addressing these issues. How can we manage the Popum arboretum efficiently as a self-sufficient venture as well as a high scientific enterprise? And also, we are trying to see whether forest cover in Sri Lanka can be increased by this method, assisted natural regeneration, which was started by Popum in the 60s, in the 1960s. And then you want to see what kind of plants we have in this country, the flora of Ceylon, flora of Sri Lanka, and how can we use some of these plants for economic benefit, sustainable use, and also the factors affecting the conservation of these very important indigenous flora. Of course, we started with upgrading the Popum arboretum. This was the original cottage occupied by Mr. Popum back in the 60s, and we upgraded that. And also we erected the fence right around the garden, right around the arboretum last year, and also put up various signboards showing the different components of biodiversity: birds, butterflies, bats, mammals, and assisted natural regeneration, also information for the benefit of visitors. And some important plants were also provided with interpretive signage. And then we were getting a lot of school children, and we were conducting programs, day tours, and various things. We will get these school kids. These actually, some kids came from Ratnavati Balika Vidyalaya. A lot of people who came and we conducted a very interesting program. And there were a lot of people who are coming to study foreigners, local people, and these are group who came to study fungi. And also, as usual, there are people coming to watch loris at night. We are conducting loris safaris. And as you can see, over the years, the visitor numbers increased. Actually, we started to keep records of visitors starting from 2005, when the management was handed over to the local. But then, as you can see, there's a marked difference in the visit numbers in 2017, because Chanaka Lekam we were there, because we started resuming the management committee, and whether these were not documented properly or whether the numbers increased, still not sure. But as you can see, there's an increase from the 2016, 17, 18, in spite of the bomb blast in 2019 April. There was only a slight dent, but unfortunately, in this year, after March, no visitors. Chanaka is working on the restoration ecology. He was studying all the plants about 10 centimeters at the Arboretum, and he found that there are about 369 species, and he found, comparing the previous data, that there's an increase of canopy trees and sub-canopy trees, so that the Arboretum is moving towards the climax vegetation in the rhizome, the primary forest. And this is the take-home message. Now, we are doing fundamental research. By the way, taxonomy is the most fundamental branch of Bali. So, we are also providing some solutions to various problems. Like, these are the 3 species we are suggesting to the forest department to use as reforestation. Reforestation. Instead of higher eucalyptus 26, why not use liquid and vellum? So, we have found that these can be used in place of those exotic plants. The other student, other research assistant, Chanaka Lekanga, is working on the Jambu family, and he's working on molecular and morphological aspects of the genus. He has spent 3 weeks in Singapore studying molecular techniques, and he has even found several new species which are to be described very soon. There's another RA, research assistant, she's Dilanti Rajapaksa. She's working on another interesting genus called Strobilanthes, commonly known as Nelu. I'm sure in all places you have seen beautiful flowering or blooming of potentials, which happens earlier. So she has also done several studies, and she has worked on polar morphology using electron microscope, and she has also described found a new species, and papers ready to be published describing this new species. Last year also I presented data of Aishani Samarasinghe. She's also working, she's the lead investigator is Professor Bandar, who is here. She's working on invasive alien species and trying to extract chemicals which can be used as agrochemical. Now, last year I presented data on antifungal and anti-insecticidal activities of some plants. And this year, last year actually she found so many antibacterial compounds from these invasive plants, and Professor Bandar and the team are even planning to write a patent application. There are 3 other graduate students who are working not attached to IFS or in the University of Peradeniya. Tenafon actually the head of research at Link Natural Products. He submitted his thesis last year, and also Taranga Vijayvikram, he worked on Hatuna in Moragaha. He submitted his thesis also, and Samantha Gunasekara, formerly of Sri Lanka Master Assistant, doing his PhD at Ashoka University on Landa. And there are 3 undergraduate students who worked on. They are all from Sabaragamu University. One worked on Ulex europaeus in Horton Plains. The other one visitor perception on invasive plants in both Horton Plains and Udavalle. And Mihiri also worked in Udavalle on the life after Lanta. In addition to those, there are a few other activities we conducted. 5 public talks were given in various fora in Colombo mostly, and also one school science or education program for school children was carried out. And I gave a 4 invited presentation and 2 workshops, one on lichens and one on fungus was conducted. And there were 2 papers, SCI papers published last year, and 2 SCI expanded papers were published, and one of them is actually describing a new species, new high plant species. Describing high plants is a very rare, not that many species have described in the last decade. And then there was one peer-reviewed journal publication on Madan. And of course, there were 19 abstracts published last year, and we are planning. These are the future. Actually, these future because last year I presented. We are working on the arboretum development because now arboretum is ours. We took over the management from 1st of May this year. We are trying to continue. We are trying to develop this place, as I mentioned earlier, and then taxonomy of various groups, angiosperms, high plants, ferns, lichens, and fungi. In fact, I had some consultation with Chinese academic sciences, and they were willing to fund a project on flora of Ceylon. That was actually after we finished the Smithsonian project and we showed the project. So, the first time we are getting Chinese support from Chinese academic sciences to devise the flora of Sri Lanka. And some other scientists in China trying to help me to study fungi. And also, now we did, in fact, we are publishing red list of plants. Now we are into red list of ecosystems and regeneration of forest we are continuing with these new Sri Lankan plants and sustainable use and also research on invasive species. Okay.
The presentation is open for discussion. We have 2 minutes. Yes.
What Papam did was to remove the undercover, and then he said that the trees will grow better. I think to grow a forest, that may be a good thing, but do you think that the question about the carbon footprints, the natural forest with the undercover will capture more carbon compared to the type of things what Papam is doing? I think it is good for to have good trees to get the products or timber and things like that. But when it comes to the carbon footprints, doing that won't be a good thing, I presume.
No, I think I have to clarify. Now, what Mr. Popov did was removing competitive weedy shrubs. Now, the natural forest seedlings took off and then they provided the cover. Now, after that, there will be other natural seedlings that will come. So, we are not going to remove any of those natural shrub species. Now, when you have, I think that gentleman who presented the keynote address also mentioned about the functional diversity. In any forest, there are various components playing different roles. Like if it's shrubs, as you mentioned, canopy trees, subcanopy trees, animal, all sorts of different components of biodiversity are playing roles, functions, functional diversity, higher functional diversity in the climate forest. Now, we are not removing any of the native shrubs. We removed, Popam only removed the weedy, invasive, competitive, exotic shrubs, not the native ones. We are not removing those.
The weeds are also very important when it comes to photosynthesis.
Well, when you say weeds, definition of a weed is the plant which is in the wrong place at the wrong time. But in our case, it's not a weed, if it's a native herb or a shrub.
What I feel is, moment you remove these things, you are deviating from the optimum photosynthesis.
You will review the competitive exotics, not local plants.
Yeah, unfortunately, we have time constraints, although the discussion is very interesting. So we go move on. Thank you very much for the listening. So we have the next presentation. That's on plant stress biology and molecular genetics by Professor Saman Senagiran. Professor Saman Senagiran does not require any introduction. Professor Saman Senagiran.
Mr. Chairman, and this is my second role. I'm a scientist as well. Today I'm going to talk about the project I'm working on, plant stress biology and molecular genetics. And I had, in fact, my presentation was done this morning, but it was not there. Like I had to adapt the last yesterday version. Actually, this I have missed few bits. So overall, like I have 4 projects I'm running now here. So one is dissecting the genetic mechanism of rice. It determined the yield potential. That's working. I got funding from Treasury, and also working on another project to understand the fundamental mechanism of how nutrient transfer into the grain, rice grain, and aiming to improve the quality, because this is one of the biggest problem in the not only Sri Lanka, as globally. And the third project I'm working on is to improve the nitrogen use efficiency. And still, I think nitrogen use efficiency, though we have discovered all the new technology, nanofertilizers, we are far behind, because the biologics and the nanotechnology need to come up with a hybrid, to understand these two together. So this is where I see a big opportunity. And at the same time, I got later on in my career, got interested into the human element conflict, and we got funding from the NSF to do a project and to develop a new technology-based solution to manage the human element conflict in Sri Lanka. So we'll jump into the first project. I will quickly go into the story behind. So this graph shows how rice yield potential changed over the years since 1978 to today. So its yield increased and now start to plateau. Its current yield is somewhere around 4.2 tons per hectare. And as said the secretary in the morning, that this is the country population, nearly 60% is around this industry, and it's a 4.2%. The yield is not good enough to. It is okay. We are sustainable. We manage. We are self-sufficient. But at the farm gate level, at the farmers level, they are economically not sustainable. They don't even make a break even. Because of this simple reason, we need to push the yield gap at least to up to at least 6 tons, 5 or 6 tons. That we need to do quickly, otherwise people get out of this industry. And so what we hypothesize is there are places like, for example, in Australia, the yield potential is 17 tons per hectare. But in our is 4 tons per hectare, and in Japan, 10 tons. China, maybe another 10 tons. The US, roughly 8 tons. So why we miss? What mistake we have done? What we hypothesize is that during the breeding programs, during the whole after the Green Revolution, we did our own breeding programs. We missed some of the traits, that biological traits that require to push the yield potential. I think we miss out. We need to remine our rice genome to pick up those traits that we miss, and we need to reintegrate into our rice genome, and that's I think is the most important thing. So we propose. We require to, one thing is to set up a rice co-collection, world co-collection here in Sri Lanka. That never happened, but this is the first time that we collect the varieties from global, the significant, it has big yield variation, and include our own varieties and accessions and so on, and see the genome behavior. And so we have already started this project here, and we got the agriculture department involved, and also Biodiversity University involved, and so that's the selection is set up already, and we have started phenotyping and genotyping, and also we do this work with one of the Japanese university, and they're helping in genotyping work, and also we do transcriptomics and bioinformatics. We bring these things put together. And again, this is a very early stage. Just last year we started. And so once if you identify the promising traits, genetic traits or phenotypic traits, that has a direct link to the yield. So we are also ready to do genome editing to get the yield potential up in Sri Lankan system. So what we want to do in the next 4 to 5 years time, so current yield is this is a 4.2 metric tons, that we want to lift it to the 6 metric ton. That's if we can do as institute I think we have done the job for the country. This is, but again, we need the national support. It's a very difficult to do. It's not a single man job. It's not a single institution job. It's a multinational institute, and we need support from the global support as well as the internal support. And finally, what we want to do, we are in the process. We want to develop a plant that have idiotype as a more pentacles, more grains, more more mass or yield mass compared to the shoot mass. This is what we are, but can be done and can be engineered where if required, and it is possible. It is happening in the other part of the world because we don't have that technology. That technology we need to get here and we need to re-establish here to achieve that target. So the benefits will be billions of dollars to the country. So the second project is working on get the micronutrient is a grains like what we like in most of the cereals now we bred for the yield, but they have totally ignored the micronutrient. Micronutrients in terms of health play a massive role that is being heavily depleted. This is something I want to show. Most of the micronutrient coming from the cereals and pulses, and in the rice, in this part of the world, our diet is 70% of diet is a rice, and the nutrients mainly coming from these cereals. And the depletion of micronutrient that's heavily depleted now is have huge impact on a number of metabolic diseases. So I don't want to go into the details. And luckily, when you see, I use the two plants. One is wheat. I do that because as a model plant, because we need to have both understanding to do this job. This work I do in Australia because this is a genetic variability. And the right side here is how nutrients accumulate in the grain, and there is a large genetic variability. At the same time, this is rice, in the Sri Lankan our rice, there is also a genetic variability. This shows us the very good that the information is there. We need to precisely detect this information and we need to get into our breeding program. So now, so far, we have really characterized how this nutrient taken up by plant and the genes involved in the process, and also like how we transport and how load into the brain. And most interesting stories, we just completed this work with the Australian synchrotron facility and using a fluorescence microscopy, like a X-ray fluorescence microscopy. It's like a similar to the CERN in Australia, and where we have characterized where this zinc is located, and at the same time the mechanism is lying here here. The most of the zinc load in the brain through this area, and this is also called like a modified allele layer. And so we have characterized this again. This graph shows how the zinc and iron and manganese distribution. Through this process, we can clearly characterize and where it load into the grain. And I missed the genes and the markers in all. That's what I said. I revised. I missed out that bit actually. Here we have identified the genes that involve in loading in an endosperm. So now this is the first time established, and this is the first time we elaborated the mechanism of the nutrient loading particularly in the brain using a synchrotron facility. And so this provides a very promising information that future to the gene editing technology. Yeah, so we can go into we can modify the rice grain to enhance the. And this I will quickly go through the. And so next project is working on the nitrogen use efficiency, and this is like again, as I said, a lot of people working on the nitrogen use efficiency, and there's no biological matching work has done. In this project, we are looking to get the N nitrogen to match the biological demand, and here we have developed a new fertilizer. It's called hybrid N that support the biological N demand of the plants. And yeah, so we also have a project in a human-elephant conflict area, and we develop a new collar, that satellite-guided technology to control elephants that we use in a Pinola elephant orphanage. And overall, this is a publication outcome of the year. 17 publications and citation around 7,000 and 5 PhD completions and 3 undergraduates and a couple of books as well as few PhDs, PhDs under supervision. Yeah, thank you.
Thank you, Professor. It's a very interesting presentation. Unfortunately, we don't have time. There are time constraints, so we are going to skip the question section. If you have any question later on, you can ask Professor Anira and discuss them. So,
technology and advanced materials. May I request the speakers to stick to the 10 minutes time, because there are 5 more speakers in this session.
Afternoon. I'm going to present the progress of our project last year. This is the outline of my presentation for you. The outcome will be presented are 2 sub-projects. This is the most important part of our project, our research team. Unfortunately, it has been limited to 4, including only 1 regular in this year. But we have the support of 3 visiting collaborative scientists and a number of collaborators, co-investigators, post-graduate supervisors locally and from abroad. The first introduction, nanotechnology and advanced material. The 2 most progressive branches in material science and technology that has already played a key role in improving the economic performance and the quality of life. As you all know, nanotechnology has already created many new materials, devices, for a vast range of applications. However, we should not forget that at the same time, our country possesses a large variety of economically useful minerals. Unfortunately, they are still exported as cheap raw materials, but they are having a high potential to play a major role in those high-tech, high-profitable nanotechnology and advanced engineering applications, industrial applications. However, proper high valuation to our minerals by carefully developing for this application is lacking. Therefore, our project of nanotechnology and advanced materials seriously mind these factors into our country when adapting or contributing to this scientific and technological advancement in nanotechnology and advanced material field. Now I will move to one by one of our sub-projects. So, first, our aim was on value addition to Sri Lankan minerals and related materials for advanced application. We start this with Sri Lankan graphite some years ago, and some of you already know that we have achieved one of the import outcome of purifying them up to ultra purity of 99.9 over 99.9, and through developing our own techniques and the processes, and we were able to get a patent for that one. And without stopping there, we further move into development use of them in different applications. One of the main applications was battery, especially rechargeable battery. That was done by through modification with our highly purified graphite through develop and introducing new low-cost structural surface modification methods. First, we developed them for the direct use in lithium-ion battery. That's mostly used at present, and also we are now presently developing them for future upcoming sodium and magnesium battery. So here you see the steps of development of Sri Lankan graphite for lithium-ion battery. While developing the material, we also developed a laboratory facility. Now we are capable of making those laboratory scale batteries at our laboratory. Out of this part also, we obtained 2 Sri Lankan patents based on the outcome. And further to that one, I mentioned about the lithium-ion batteries. Now we are working on developing them further into use for the upcoming sodium and magnesium batteries. We are expecting them to have good future soon. And then it's not a simple work. They are here to do some structural modification by through varying interlayer spacing. That's for using sodium and magnesium and other which are cells that use bigger ions than lithium. So there we found a very important achievement very recently. So they are this is not only limited to battery application. We were able to develop a new method to engineer the interlayer space of graphite. That means between graphite structure and the graphite oxide structure by developing changing the chemical composition experimental parameters. Yeah, you see our first attempt of using this one showed 360 milliampere per hour in the battery of capacity, and the most important thing is even after 30 cycles, it maintained the efficiency of 97. It's the first run of this type of battery. Actually, that's a big task every year in the world, searching for electrode materials for the sodium ion battery. I think this is human, and we are now we are preparing for patenting soon. And then our second project was mostly involved with the synthetic material. That's raw materials are from outside, but anyway, here we develop our own nanotechnical methods and low-cost methods and our own laboratory development for the use of this material. Our strategy was to develop transition metal oxides with cheap additives and introducing nanotechnical methods. There also we found we developed nanostructured sodium titanium oxide. That's a famous anode material for sodium ion battery. And there also we found some interesting results, but this work is still continuing. And apart from those things I presented so far, our research outcome, apart from that, we have spent a lot of time and energy in the recent months and year by developing a National Center for Advanced Battery Research attached to our laboratory at NIT. That was done mainly funded by General Treasury of Sri Lanka under major grant which is good work has been paid. This is not only linked to NIT, it's a central laboratory facility open for all local research groups working on developing local minerals for battery and energy-related application. You see the pictures of the ceremony opening that took place in January this year. So, and at the moment, our facility, laboratory testing battery ranges from material synthesis, component fabrication, cell assembly to battery performance evaluation. But anyway, still we give the highest priority for of the center for development of Sri Lankan minerals for this type of energy-related application, especially for battery application. So now I move the progress. As I mentioned, we have spent a lot of time on laboratory development in the recent months, almost 1.5 years. At the same time, the grant we received was drastically reduced. Even from the outside grant, it's down to 25%. Actually, we were struggling, and even with that, we could maintain some reasonable progress in the last year while we were in laboratory. Thank you.
The presentation is open for discussion.
If you do develop these batteries, how do you plan to further make it a marketable product?
Yes, actually, sir, our first product was that highly purified graphite, then the battery-grade graphite. Actually, we are in a position now, patented battery-grade graphite. And last year, actually, at the beginning of this year, our institute called expression of interest to a newspaper advertisement, and some investors came. And just before the COVID pandemic started, we had the first discussion, and now they got our information, and now it's under discussion. And also, out of that, some other investors also came, something with the international linkage, and all are under discussion.
Thank you.
In the absence of any other questions, shall we go on to the next one?
next presentation is on microbial biotechnology. The presenter is Professor Gandhi Senegal.
Thank you, Chairman. Right, what I would like to do is to go through this sort of summary thing and to show you a sometimes interesting video about the project. Right. Overall objective, application of microbial biofilms, biofilm technology to solve various issues in agriculture, plantations, and the environment, and also to produce biochemicals in various applications for various applications. Specific objectives for the year under review: basic research investigation to study the potential of biofilm biofertilizer application in paddy and tea soil carbon sequestration in Sri Lanka. Is already explained in the annual review book. Therefore, I'm not going to go into detail to save time. What I'm talking a bit more about is applied research aspect, large scale rice field trials done during March 2019-20 season in collaboration with the Department of Agriculture to examine the efficacy of biofilm biofertilizer. These trials are large scale in 25 locations, 5 districts in each district, 5 locations. In each location, we had about 1 acre, 3 consecutive compound plots. There are real farm fields, large fields, 1 acre. Districts are Anuradhapura, Pavanagar, Kedwal, Kurunagal, and Ampara. Total trial extent was about 75 acres. Treatments, department recommendation, then 65% of the department recommendation, which is equivalent to about 50% of the reduction of farmers' practice because farmers use a high amount of the recommendation. Then again, 65% chemical fertilizer plus biofilm. We use only 1 liter of biofilm biofertilizer per acre. It's very convenient here. Results, all locations saw that biofilm biofertilizer used with 65% chemical fertilizer, that is 50% farmers practice, gave 10 to 30% yield increase over the department recommendation. And in 70% of the locations, the yields of the biofilm treatment were significantly higher than that of department practice, and also our check on the control reference treatment 65% alone. That is farmers 50%. So this clearly shows that biofilm is not a trivial thing. It can really significantly improve rice production in the country. In conclusion, biofilm biofertilizer can cut down farmers' fertilizer use by 50% without hampering yield, instead increasing the yield on average to 20%. Biofilm biofertilizer can reinstate sustainability, price of fertilizer system, economical and benefits, and also environmental benefits. Last year, as our Secretary of the Administration, the Colombo Declaration was signed by Sri Lanka. There we agreed to cut down nitrogen waste by 50% by 2030. So biofilm biofertilizer will be an important, very good candidate to achieve this target. And also the recent news, Cabinet of Ministers has now approved promoting biofilm biofertilizer for paddy cultivation island. So that's very pleasing news for us. Our team last year, Project Usura and myself, 3 research assistants, technical officers, 2 technical officers, Ministry project volunteers, because this grant is work is funded by the ministry. Co-investigators, collaborators, University of OLS University, whatever. Right, what I would like to do is to this video was made for farm organizations as well as agriculture officers up to field level. Therefore, this is example. So we watch the video.
ජාතික මූලික අධ්‍යයන ආයතනයක් මගින් ආරම්භ කරලා තියෙන මේක සත්‍යත්වක තාක්ෂණ හා නවෝත්පාදන අමාත්‍යාංශයේ යටතේ අපි ආයතන විසින් මේ පිළිබඳ පරිශන ආරම්භ කරේ වසර 15කට විතර කලින් ඒ සොයාගැනීම කරලා දැන් පේටන්ට් බලපත්‍රයක් සහිත ලෝකයේ පළමුව තාක්ෂණිකාවේ නියමිත සංකල්ප විශේෂයෙන් රටේ නියමන සඳහන් කරනවා. विशाल वर्ष सौख्य संपन्न जयपुर के द्रव्य उन्नत एक विभागावे में वीर इसलिए कि इसी का आवश्यक है विशेषण तो द्रव्य लेकर बताया लेकिन द्रव्य या किसी सिंगल प्रमाण लेकर सूत्र प्रमाण लेकर जो आत्म निश्चय करना है रिलेवेंट लेकर पूरा वाला एक रिलेवेंट में उसके पोटेंश और निश्चय करना सामान्य और आकार है ऊपर इस वाक्य में कहते हैं कि तेरे इसे जो मतलब ये एक संपूर्ण अपाय होते हैं एक वाणिज्य में वर्ष निष्पादन के लिए मतलब राज्य बाउंड्री का सारे बताने के लिए सीएससी देशीय समाज संघातन में कमाने की निष्पादन करने वाले राशि से गाना काटने उतर कर पांच दाल करने के साथ हमारा विस्तार करके दुनिया पर करेंगे
විදර්ශී ශාකාලයේ කුඹුසලම විදේශ සමාජවල ලබා දීමේ වැඩසටහන යටතේ රසායනික පොල් සමග බයොෆිල්ම් දෙවකුණ ලබා දීම සිදු කළා. ඒ යටතේ පරිපදේශයේ කුඹුල විද්‍යාලය 1965 සඳහා බයොෆිල්ම් දෙවකුණ අපි ලබා දුන්නා. පසුගිය කන්‍යාස සිදුවීමේදී ශ්‍රේෂ්ඨ පරීක්ෂාවේදී කුඹුල ගුණ සපලකට ලැබෙනවා. ඒ වගේම තමයි ගොවේකී මේ සඳහා තවත් ප්‍රචාරණය ලැබිලා තියෙනවා සාර්ථක බවට. ඒත් ඊළඟ කන්නේ සඳහා තුනේ මේ සඳහා වැඩි ඉල්ලුමක් තියෙනවා. සාංකේතික රසායන පොල් විතරක් භාවිත කරලා වැඩ කරපු කුඹුරටම්බල මේ දකින වෙනසත් එක්ක මම හිතන්නේ ඉදිරියේදී නියෝගව සඳහා යහපත් කියනවා කියලා මට මේක ප්‍රකාශ කරන්න පුළුවන්.
මේ වගේ කුඹුර දැන් මේකේ වැඩිමද පාත්‍රවල වැඩිම කඳේ වැඩිම පළල් භාවය ඒකේ සංසාර භාවය මේ කුඹුරේ ඇවිල්ලා තියෙන අඟල් භාගයක අඟල් විතර තියෙන කපුල එදාගෙන යන මඩ ප්‍රමාණය දැක්කම මට තේරෙනවා එදානම් කුඹුරත් එක්ක පරිසරත් එක්ක ඉයතුන අපිට මේ කුඹුරේ පොඩි වෙනස් වීමක් කුඹුරේ සිද්ධ වෙනවා ශරීරක මගේ සෞභාග්‍ය සංකල්පය යටතේ මේ සංසාර රටක් එක්කලා වසතියතර සමාජයක් රටක් බිහි කරන්න තමයි බය නැහැ. මේ දුල කියන අර බයෝෆිල්ම් ඒ පියර මේ පොර එකයි පාවිච්චි කරලා මේ වගාව කරගෙන යන්න කිසි බයක් හානියක් සමාජයේ අස්සන්නට අවසාන ප්‍රතිඵලයට හානියක් වෙන්නේ නැහැ.
සාමාන්‍ය වසරකට හෙක්ටර් වශයෙන් ගත්තොත් මේ වඩාම ඒ ප්‍රමාණය අපි ගණනය කළොත් වසරකට දැන් රජයේ වැඩ කරන සාමාන්‍ය කාර්යක්‍රමයකට ගත්තොත් බිලියන දෙකක් රජයට පුරවන ඒ වගේම ඕවර ආමේ කිරීමට නිරන්තර විදේශීය ජීවිත බිලියන 6ක් රජයට පුරවනවා. Esse marca de abertura que eu tenho, eles fazem lembro da gente que trabalha os dados que está acontecendo eles dizem que quando está verificando aquele classe se a intervenção de que eu tenho que botar aqui sobre a coisa de ter a ação que eu sou da coisa porque eu it was already put out the
Thank you. Thank you, Professor Ratna, for that excellent presentation. Because we have gone over time, we would have time for questions. We have to proceed for the next one. But I think the most important thing, policy impact, you mentioned for the country. So let's proceed to the next speaker, Professor Veera Surya. He will be presenting to you on environmental science research program, water quality.
Good morning. So, on the environmental research program, a lot of national and international institutes. So, environmental water quality research is based on this statement given by science journal recently, running out of time and running out of water. And UN has 17 goals, and we suggest these 16 goals cannot be achieved by reaching the water goal achieved. So we put the water as a central and rest of the other goals surrounding that. So during the last year, we were working on several programs. One is drinking water desalination, and what is in green, we are going to make presentations today. Then development of water quality index and electrical water treatment. And we just opened up the National Center for Water Research, and I was questioned, and our group was questioned, why there's one more center when there's over 25 centers on water research in the country. So this is Sri Lanka. We are abundant of water. Still, about 4 million of people are without water, without safe drinking water. At the same time, downhill, we are having Kongan now. We are wasting good quality water in the country to wash the cars. So I think for this component of the project, we got funds from National Research Council, and we come together with several institutes in the country. We try to solve these 4 million water quality issues. So if you think of the solution procedures the government of Sri Lanka is forwarding toward, none of the systems were, I would say, sustainable. And we are doing a lot of incorrect scientific methods for doing these things. For example, the rainwater harvesting systems. Presently, we are doing the rainwater harvesting in the downs where we are with a concrete tank. But in the ancient time, in Parakramba, harvested the rainwater through the reservoir system, which is very scientific method. So based on that, this is very famous inverted pyramid, which is very special to IFS. So based on that, we define the water quality issue in the dry soil is a fundamental chemical research problem. So our water problem is due to the drinkability or palatability issue, and we are giving the solutions which are not accepted by the community after time. It is not that sustainable. So IFS, we believe these concepts first.
Our first argument is natural water cannot be synthesized. That is number one. So we have to protect that. Number two is, so how can we protect that? So we have to go to the fundamental science here, and we want to define some index for the protection, and we have to improve the palatability. So, and so far in the country, when we are trying to solve this problem, we have forgotten very important issue, that is natural water cannot be created synthetically. That was forgotten. So that is the reason for the water problems in the country. So our solution came this way. Because research results usually take time to complete and get a success. So first we will do a solution. So once you do the solution, why there's a research is needed? Solution is interim, which is from the available technology we use to apply correctly to solve the problem. While we are solving the problem, we do the research to attain the sustainability. That is the argument we are making on. So, number one, this is very important side because this gives a lot of social problems we do have. Because for this project, we do the research part in parallel with the solution part. Okay, for the research part, for the solution part, we wanted to select a village in the dry soil area. Okay, at the same time, bottom line, we say Rajarat University research. That is the research part. Both are in parallel. And we took over 2 years or 3 years to select the site for the location. For example, in 2017, we were nearly escaped from the people by going because people try to attack us because they have no confidence on us. Okay, like that we had a lot of problems. Finally, we selected what is in the middle, one village site, which is a poor school, primary school, and this is the site we selected. While we are having 3 years of struggle for the selection of the time, and we developed the water facility at Rajarat University. Okay, so we believe that we should not take villages as test sites. It is not permitted. So we developed our site in Rajarat using the abandoned well, which is not used. So these mechanisms are there. And number one, site selection, we selected this village. This is very beautiful geology. My colleague, Professor Dipal and Dilshan solved this problem. This is the road. One side is our side. The other side is there's a water plant offered by Navy, Sri Lankan Navy. But it is a dry well. It is a geological problem. And this group helped us to get the correct location for the water site. And it is a heavy geophysical studies they did for us. Then we constructed a model treatment facility within a 1 year's time. It is right below now, and the Secretary of Ministry of Urban Development is visited this place. Now this system is in operation in this place. So now I told you that we are going research in parallel with this. So our research part in parallel, I am discussing, this is the water quality index that we are using today. There are so many elements we have to check whether the water is good for drinking or bad for the drinking. So this is very problematic. So we decided to have a one number like the GPA of a student. From that we can tell whether the water is good or bad. That is our intention. Okay, so with this aim on mind, and here I put in red, basic science goes unclaimed generally. What we are doing, very hard science is not never mentioned. That is the nature of basic science. Here also, we have to go for this from the right corner, because once the element is in the environment, how it behaves, we call it mechanistic model, how it behaves. But it is a prediction without looking at that. So then we want to go to the quantum mechanical calculation, whether these predictions can be assured but that is not enough. We have to observe it. For that, we have to go for the site control facility from which we can identify the exact chemical species which is available in this water site, exact biologically active chemical species. Rather than having a lot of these species, we got this identified partially. And because of that, we suggested this development is first version. And this is, I would recommend, this is the second study that we did in USA. It is partially successful experiment because for the, I think, very rare experiment, we do synchrotron for the solution phase. The whole idea is how the fluoride and the aluminium occur together, the biological activity. So this is a bit hard question, but our results are not that good. We have to repeat the experiment. Because of that, with the help of our statistics department at the University of Peradeniya, with this uncertainty, we developed somehow an index, collated all the parameters. Now we have one number to get whether the water is good for drinking or bad for the drinking. So based on that, our statistics group in FIFS developed a very nice map covering the whole region, where red indicates the bad quality water. And this is the first time, I think, the water quality index is spatially distributed. They use the technique called, spatial geostatistics. Because of the time constraint, I will skip these things. And it is very cheap. Our system is 400 rupees for 1,000 liters. And these local talents were used for the operation. And I will skip everything because of the COVID, we had positive and negative impacts. And our social outreach program was very intense. And this was organized by National Research Council, and it was delayed because of the COVID pandemic. And what is red? We say water is deceptively simple molecule, and that is the reason for the Water Quality Research Center at IFS. And with that, we conducted a lot of outreach programs. We called Water Has a Life, and this is the thank you meeting given by the students. Thank you.
Thank you, Professor Veerasurya, for your interesting presentation. Unfortunately, we don't have time for question or discussion. So shall we move on to the next one? Next presentation is on Earth resources and renewable energy by Professor Deepa Subasi.
Thank you, Jaya, and good afternoon, ladies and gentlemen. In my project, actually, this project is focusing on exploration of new natural resources as well as to the utilization of the available resources, especially energy and energy minerals, in a more efficient way. Under this, we have 4 sub-projects. This geothermal resource mapping is the flagship project, which we are focusing on mainly. In addition to that, there are 3 other projects which are quite deviated from this main project. So I will go through each project briefly. So the geothermal resource mapping project was initiated by Professor Disanayake, and the main idea is to identify and explore probable geothermal areas and also to evaluate them with view to utilize them for national development. So this is basically IFS funded project. And the second project is thermoelectricity project, which is basically a material science project, and it is partially funded by the NRC. And the third one is the evaluation of natural resources in Sri Lanka, and it is in collaboration with the Department of Geology, Dr. Malaviyarachchi and Dr. Dharmapriya are the main investigators there. And one of the projects is funded by NRC, and the other one is funded by the Ministry of Science and Technology. And in addition to that, there's another radon mapping project which was started as a part of the geothermal resource mapping project, but later it was developed into a separate project. So in this project, we try to evaluate the geothermal resources and to find out new geothermal resources. Currently, actually, I had two students, both of them have left now. One completed his MPhil and he just completed his MPhil. The other one has gone for PhD. And so the main objectives are to produce a geothermal resource map and investigate the origin of the heat sources and investigate the Highland-Vijayan lithological boundary, which is a geological boundary, and it is connection with the thermal springs, because most of the thermal springs are loosely scattered around this boundary. And we have main local collaborators, Geological Survey and Mines Bureau. And we have several geothermal springs in Sri Lanka. And of course, if we can develop these geothermal springs into produce electricity, they will provide clean and renewable energy. And also, they are quite versatile because unlike solar, hydro, or wind power, they do not depend on the weather conditions. And in this investigation, we use geophysical techniques such as magnetotelluric, time domain electromagnetic resistivity, and also geochemical techniques such as measuring chemical and physical parameters, analyzing dissolved minerals, and also isotope studies, and also geological mapping. And this is actually an output of one of the geophysical methods. So this is near surface. Actually, with magnetotelluric, we could go down to several kilometers. So these are showing the resistivity profiles, which are actually can be interpreted as the flow paths of the water or structural features. So, so the future directions, we actually want to mainly develop this into energy production, electricity production, but that has to be taken up by the engineers, and we cannot go, but we can provide all the information data we have narrowed down where the production can be done. And also, we can use the mineralogy and petrology to further enhance the understanding of the lithology, and also a satellite image and data analysis can also help to narrow down and understand. And second project is this mineralogy and petrology Sri Lanka project. Dr. Malaviyarachchi and Dr. Dharmapriya are collaborators, and research students, we have two research students, Ms. Nayak and Ms. Gayatri Vijayaratnam. And here also, it's mostly focused on basic sciences, but we also hope to find new mineral resources, and they can contribute to national development eventually. So I'll go into the next one, thermal project. We have one research assistant from the IFS and one from NRC grant, and which came to end recently. And we have one volunteer, and this thermal electricity can convert heat energy directly into electricity. And so it can be used to harvest our scavenge energy, and also it can be used as a renewable energy source. So we are mostly doing theoretical work as well as we are trying to develop material and also some thermoelectric modules to generate power. So they can have a lot of applications, especially space applications and niche applications. And this is a theoretical model. And actually, in addition to theoretical model, we developed this small mobile charger, which is working on a small candle, and it can charge a mobile phone. So this is very useful when we don't have electric power, or when we are out stations or camping or something where we have no access to grid power. So this one I will skip at the moment because of the time strains, and we started to produce radon mapping using indoor radon detectors as well as outdoor radon. The previous one was indoor, and this is outdoor radon concentrations. They can be used to identify the mineral resources, especially uranium and thorium mineral resources. As well, also we can understand if there are any high risk areas. And we have several publications and conference presentations, and there are some awards, and I was the president of the Geological Society, organized several conferences and training programs, and conducted some keynote addresses and plenary speeches. And so we have several grants. And last year, 2 students submitted, and 1 student got his MPhil degree. And recently, another student completed his MPhil and submitted. So I like to acknowledge our awardees, awarding agencies, NRC, NSF, Ministry of Science and Technology, and also the director and the staff of the National Institute of Fundamental Studies and Department of Geology, Geological Survey and Mines Bureau, and all other collaborators. And last but not least, I want to thank my team. Mr. Padan, they are the technical officers, others are students. So, and thank you very much.
Thank you, Professor Kusinga. Unfortunately, we don't have time for discussion. So let's proceed to the next.
The next presentation is on bioenergy and soil ecosystems by Dr. Renuka Ratnayake.
Good afternoon, ladies and gentlemen. Bioenergy and soil ecosystems are the research areas under investigation. I will first briefly explain the progress of the bioenergy project. 2 sub-projects were carried out. One is microbial cellulases, the potential applications in biofuel production. Cellulases can be utilized as biocatalysts in industries, and normally genetically modified microbes are used for the extraction of commercial available cellulases. This process is expensive, and it is important to utilize my natural resources for extracting cellulases. in this project, we studied the possibility of utilizing enzymes extracted from naturally occurring microorganisms to replace the use of commercially available enzymes. Isolation of bacterial and fungi strains were carried out using soil samples, and isolation of anaerobes were carried out from termite gut and also from rumen fluid of herbivores. Isolation of yeast were carried out using locally available fruits, and then these microorganisms were co-cultured, and co-cultures were detected for sugars and alcohols produced by using HPLC. From in total, 163 fungal strains were screened for cellulase production and found 3 isolates were efficient in producing cellulase. And total 54 bacterial strains were screened and found 2 bacterial strains were efficient in producing cellulase. And from anaerobic microorganisms, 1 strain was effective. And these strains were tested for, individual strains were tested for ethanol production. And then finally the co-cultures were screened for alcohol production and found 2 co-cultures were efficient. And we concluded Trichoderma viridae and Candida parasiticus and co-culture and Trichoderma viridae and Candida tropicalis co-cultures were effective in producing bioethanol production. The next project is investigation of genetic diversity of cyanobacteria in different extreme water bodies in Sri Lanka with their taxonomical identification, nutrient profiling and toxin analysis. The cyanobacteria are photosynthetic microorganisms rich with nutrients, and they are abundant in the water bodies. And these are some of the applications of cyanobacteria. Objectives are to taxonomical evaluation along with morphological and molecular identification of cyanobacteria, to investigate their potential industrial applications, and to establish a cyanobacteria culture collection. Water samples were collected from extreme environmental conditions such as hot water springs, lagoons, mangroves, and salt marshes, assuming that the organisms found in these ecosystems are effective. 200 different microorganisms were isolated. 75 monocultures were from freshwater bodies, and the rest from the extreme ecosystems. From these isolates, we found another strain from water bodies of Jaffna, and recently published in the SCI journal. Experiments are ongoing to study the suitability as a food substitute. Next project on soil ecosystem. The aim is to collect baseline information for soil carbon, estimate soil carbon stocks of natural and agricultural ecosystems, and to prepare GIS-based map to show carbon stocks. Carbon is the most important indicator of soil fertility, and storing carbon in soil is one method of reducing carbon dioxide from the atmosphere. So by this process, carbon credits can be earned. 2 sub-projects were carried out. One is development of baseline soil information system for soil carbon and other nutrients for paddy growing soils of Sri Lanka. And waterlogged conditions and anaerobic conditions in paddy soil reduce the decomposition rates and thereby increase the carbon sequestration potential. So the paddy has a higher contribution for carbon sequestration and carbon dioxide removing from the atmosphere compared to the other upland crops. Main objective is to develop a digital map showing the contribution of soil carbon stocks and other nutrient cations for paddy growing soils of Sri Lanka. Project outcome will be used for the establishment of the national carbon accounting system and also for the site-specific fertilizer recommendation. So these are the sampling sites all over the country. 1,600 samples have been collected and analyzed so far. And from the results, it's obvious that wet zone soils contain more carbon than the dry zone soils. The other research is soil carbon sequestration in Candian home gardens. Home gardens are the important ecosystems in residential areas. Those are the only remaining ecosystems that can capture carbon dioxide from the environment. So sustainable management of home gardens to improve carbon sequestration is important. Candian forest garden is a traditional home garden system, exists in the central part of the country and represent a perennial cropping with practice for more than centuries. These gardens are rich in plant diversity and rank top among all man-made agro-ecosystems. Objective was to study soil carbon sequestration potential in home gardens with different species composition and to compare carbon sequestration potential with the forest in the same zone. These are the studies. And we found the Candian home gardens, two types of Candian home gardens show the highest soil carbon stocks compared to the home garden 3, which is incorporated with spice crop. The total soil organic carbon sequestration potential of home garden 2, the traditional home garden 2, was comparable to the lowland rainforest and higher than the montane forest. Conclusion, the home gardens that mimic structure of and diversity of forest have resulted in higher carbon sequestration potential. And these are data will be used to develop a model home garden system in future. Project output, we have published the first digital soil carbon map based on Marcus Forest research published in a reputed SCI journal, posted with degrees awarded and 2 degrees awarded and 2 submitted. 4 publications, 1 book chapter and 15 abstracts were done. 1 PhD students and 7 MPhil students were trained and 4 undergraduate as well. I would like to thank my co-investigators and collaborators. Also, the grants awarded by NSF and NRC, and also to my research students. Thank you very much for listening.
Thank you very much for your interesting presentation, Dr. Naika. We have time for one question, I think.
Candian home garden is a minuscule compared to the ecosystems in the whole world. So how much will it contribute to the global carbon footprint by this Candian home garden?
So actually we've taken it as an example. Yeah, to model our. Yeah.
So can we implement it elsewhere as well?
Actually, these gardens are existing
within Sri Lanka, not in Candian other places.
Yeah. Yes, intermediate zone specifically, not in the red zone. Intermediate zone and in sometimes the dry zone as well.
Even in dry zone?
Yeah, yeah. So there are different types of trees we can select. Yeah.
Thank you. Thank you very much, Professor Murray.
So the next speaker is not so he is not coming. I think it was an extremely good session. Let me thank all the speakers for their high quality research. But I have one constructive criticism, don't take personally by anybody. I think it's extremely important for us to keep to the time. Most of the speakers went over time, and therefore we couldn't have the discussions. I think, discussion is a very important part in scientific presentations, and we have to ensure that time is given, otherwise the whole spirit of scientific presentation is lost. So let's take that message, take home from next week. I would have been very strict in my general sharing of international meetings, I would have cut off the microphone. But this is the first time I'm chatting. So let's, from next time, we'll stick to 10 minutes stop and 2 minutes discussion. I have been asked by the organizers to cut down on the time for lunch because we are running behind time, 30 minutes. So it's 1:30 now, gather back by 2 o'clock. Then we can cut off the time and finish on time. Again, thank you very much. It was very high quality research. But unfortunately, we have to present our great work done over many years in short period of time. Thank you. Enjoy your lunch.
Yeah.
Long past like time. Yeah, half an hour, I think very good.